Okay, well, I'd like to welcome everybody to our last meeting of January 2022. The year is passing remarkably fast. It's our fourth meeting of the IMAG-MSM Working Group on Multiscale Modeling and Viral Pandemics for this year. I need to remind you that the meeting is live streamed on YouTube and that those recordings are archived and made available for future viewing. So please be aware of that. This is a public meeting. As usual, you have our list of names. You'll notice that the masthead has changed. Reinhard and I are still there. Jim Sluka is still there, but Lorenzo Veschini, excuse the typo, Lorenzo, I don't know how that happened, is taking over as activities coordinator, and you'll be hearing a lot from him by email and otherwise as we go forward. We have our usual communication channels. Please help us get the word out, especially about our YouTube archive. There's also an IMAG LinkedIn page, the IMAG Wiki, which people are probably familiar with, and the Twitter account MSM Viral, which people can help us use. So do please follow us on Twitter and help us boost attendance and get the word out. Are there any announcements that people would like to make? I have one, which is that I'm doing a quick poll/questionnaire on usage of multiscale modeling. And so some of you may receive a brief set of questions from me asking about your usage of multi-cell modeling. And I'd be very grateful if people would respond. Of course, there's no obligation to do that. Any other questions that people have or announcements people have before we get started? Reinhard, are there any announcements related to the steering committee that you need to make?
No, I guess what I can say is that we will send out an announcement very shortly that there will be a new subgroup established on building immune digital twins. If you're interested, contact me.
Okay. Any other announcements before we get to the business of the meeting proper? Our first speaker is going to have to, before we get to that, our schedule coming up. Next week we have Hannah speaking on modeling viral co-infections. We have an open slot next week if somebody would like to speak up for that. February 10th, we have another second visit from Kevin Janes, who gave a great talk about a year ago to this group, and also from William, also a second visit. Following week, we have Giuliano Gianlupi Ferrari, who's a student in my group, who's going to be talking about some modeling that he's been doing. There's an open slot on February 17th. And then going forward, we have an exciting lineup of speakers. Please do help us suggest speakers, volunteer yourselves, volunteer your students and postdocs. Give us the names of the people you most want to hear. It's okay if you send us news items. Quite a few of the speakers over the past year have come about because someone read a science piece in The Economist, the New York Times, that they found intriguing and that they thought would be of interest to this community. So it's fine to send us on the names of people that you saw in the news that you think would be relevant to this community. So please don't be shy about that. Okay. Coming back, our first speaker is going to have a time limit. So we're going to do the first talk, followed by a brief question session, and then the second talk, and Vivek has kindly agreed to stay for the discussion afterwards. So we're going to have a slightly different format from usual. Please remember to mute your mics until when you're ready for the question sessions. And our first talk is Stacey Smith. I'm sorry, I can't do the accent. I'll try. Who's going to talk about, is the COVID vaccine likely to make things worse? It sounds like a good topic to start a conversation. So I will turn over the floor to you.
Great, thank you so much for having me. Let me just share screen. All right, so you can see my screen okay? Okay, so I'm going to talk just quickly about COVID vaccines. Let me just see if I can hide the menu. That. There we go. Okay. And so I think we all know some things about COVID-19. So I'm just going to jump straight into some interventions, which include hand washing, social distancing, masks, and vaccination. So these are, the four major things that we have at our disposal. And each of them has a different degree of efficacy against COVID-19. And if we're going to use them, we're going to use each of them some of the time. And of course, these times may overlap, right? And so there may be combinations of things. We might be vaccinated and be social distancing, or we might be unvaccinated but use social distancing and masks. And so vaccination is a thorny topic, as always. Not everyone wants to be vaccinated, and not everybody can be vaccinated. Some people are, anti-vaccination altogether. Some people are vaccine hesitant. But there are also medical issues, and there are social issues, cultural issues. There's all kinds of things that are preventing people or slowing vaccination rates. And of course, not just in the Western world, right? These are issues, there's issues of supply in developing countries as well. A lot of people would like to be vaccinated and are not able to. So what we want to do is kind of think about the issue of introducing a vaccine into a region. And of course, for most of us nowadays, we have been vaccinated, or we have the vaccine in the region that we live in, certainly in the Western world, but we might think about sort of future boosters and stuff as more variants come. Okay, so one of the issues, I think, is that once a vaccine becomes available, then other protection options often get abandoned. So this has certainly happened in many places. And this is true even if vaccines are imperfect. So, you might get a vaccine that works some of the time, or perhaps you only have one dose. And then people say, great, I never need to wear a mask again, or I can stop social distancing, I can have all the parties. Basically, I can get out of this very quickly because I have this miracle cure, right? And this might occur for people who've been vaccinated who think, well, I'm protected, so therefore I can do whatever I want, or for people who've not been vaccinated but think, well, there's enough other people, right? So it's happened in many places in the US, and it's happened in many other places as well. So this is definitely a concern. So what we developed were risk equations to figure out an individual's daily risk of infection, right? So both before and after the introduction of a vaccine to a region, right? So we sort of think of a vaccine as kind of this like fundamental change in state. So we looked at a range of 85 to 100% effectiveness, which is about sort of how effective the vaccines are. And the daily risk is modified by hand washing, which is not that effective. Distancing, which varies a lot, can be quite effective if done well. And masks, which also vary a lot, because there's a whole range of masks out there from N95 all the way down to, grandma's knitted cloth mask. So we're going to address three crucial questions. Fundamentally, could vaccines make the risk go up? Right? Not because the vaccine is directly making it go up, but could the introduction of a vaccine make it go up? If you have the choice to maximize only one thing, would you be better off maximizing the efficacy of a vaccine? So basically waiting for a good one, right? Or could you basically get an okay vaccine to many more people? What would be better there? And if we want to reduce transmission significantly, and let's say less than 50% of what it was before vaccination, how many people do you need to vaccinate in order to achieve that? Okay, so some of the factors that we use are the efficacy of the protection option, whether it be hand washing, masks, distancing, or vaccines, as well as the proportion of each type of interaction in which protection option is used. So we can use single protection options alone, right? Or we can use combinations of protection options. All right. And just for notation, I'm going to denote these by PI if they're applied before the vaccine is introduced, and QI if they're applied after the vaccine is introduced, just to keep it clear as to which is which. All right. So if we have the probability of transmission, right, well, that probability might be modified by a given protection type, right? So there's sort of a baseline probability, and then we have changed that depending on what you're doing, whether you're wearing a mask or vaccinating or whatever, right? So 1 minus beta prime is the probability of remaining uninfected. And so therefore we can basically take all the protection protocols and basically take this product here and say, well, the risk of catching COVID, right, is 1 minus the product of all of these things, where PI here is the proportion of times that a protection protocol is employed, right? And that also includes no protection. Right? So now the beta primes will be modified according to each protection protocol. So the options we have are hand washing, masks, distance or nothing before a vaccine, right? And so before there's a vaccine, then here are your risk options, right? There's either distancing, or there's masks, or there's hand washing, or there's combinations of 2 or combinations of all 3, or finally at the end, there's no protection, right? So these proportions of times all sum to 1. So the last one is kind of implicitly there's no protection happening at all. Okay, so that's the risk before a vaccine exists. And now we introduce the risk when a vaccine does exist, right? So there's obviously many more things because we take the same as before. So we have distancing, masks, or hand washing alone, right? Or combinations of those. And then also we have vaccine alone, or we can introduce the vaccine with everything as before. All right. The only thing we didn't do was include the combination of all 4 protection options. We basically thought that was kind of overkill. I mean, we could do it. We just decided to cut it there just to save a little computation. Okay, so we analyzed our risk equations using uncertainty analysis, using Monte Carlo simulations, multivariate sensitivity analysis, latent hypercube sampling, and partial rank correlation coefficients. Let me just briefly explain a couple of these that might not be familiar to everyone. So latent hypercube sampling, right? It's a way of sampling parameters from a random grid, and then you resample, but not from the same row or column. So it's got that Latin square idea, right? So it looks a bit like tic-tac-toe. And then we did 1,000 simulations. You can do more or less as you like. So here's an example with 2 parameters. So I divide each of my parameters up into, in parameter space, I say, well, there's a maximum, there's a min. I might have some distribution of them, but in this case we didn't. So we just used uniform distributions. And then I pick some random combination and eliminate the row and column. And then I pick another one completely at random, right? So it doesn't matter where I pick, but then I eliminate the row and column. And what it's doing is it's forcing me to kind of have a good spread of choices. So the parameter combinations, they can't all be stuck in one corner. They can't all be along one edge. They have to be kind of spread out, right? And if I did it again, I would of course get a different random set, but it's a random set that's reasonably well spread. And the computational cost is not so bad because there's only 4 of these, not 16. And this is only for 2 parameters, right? Well, if there was 3, it would be a Latin cube. And if there was more than 3, it's a Latin hypercube. So that's where the name comes from. And it's actually, if there's monotonicity involved, then you actually get the kind of best bang for your buck in terms of computational, cost savings, plus also maximum outcome for that. Okay, so then what we do is we rank them, right? So basically we test each individual parameter while holding all others at median values and rank them by the amount of effect on the outcome. And so if the PRCC is positive, then that means the output variable gets increased when that parameter is increased. And if the PRCC is negative, that means that the output variable will decrease when that parameter is increased. Right. So we looked at sensitivity to all the pre-vaccination parameters before the vaccine is introduced under a couple of possibilities. Either we had moderate risk or high risk. So, depending on, do we live in a high risk region or a more moderate risk region? And the transmissibility is going to increase the risk, all other parameters are going to decrease it. So here are the PRCCs. And what you can see is that the transmissibility beta is at the bottom here. So that's obviously making things worse because the PRCC is positive. The other ones make things better, but they're very different. I'll just put up the higher transmissibility. So what you can see is that the efficacy of hand washing is not a major factor. Generally, you want to be about 0.4-ish, right, in absolute value. So some factors matter a lot. The ones that matter the most here are P3 and P6, which is combinations of masks and social distancing in the absence of a vaccine. Right? So if you don't have a vaccine, well, hand washing doesn't matter so much, but masks and distancing really matter in that case. And that's kind of what we saw before the vaccine was introduced. Okay, so I'm going to look at the case of only high-risk transmissibility, because if I just go back a page, there's not too much difference between these two. So we're going to stick to the high-risk case. That's kind of the worst-case scenario. And we'll look at some limiting cases. So what if there's no social distancing? What if there's no mask usage? What if there's no hand sanitizing? So if we take out some of the options, then, well, what you see is that, okay, if I take out, for example, the social distancing, well, I'm obviously taking out any combination that has it as well. So P3, P4, and P6 are all gone. And the one that matters the most now is the combination of the other two. So it's the combination of hand washing and masks really matters. And that story is kind of true throughout, right? If you take something out, the combination becomes much more important, right? So if you can't do something, if you can't social distance because you're in a crowd, well, you need to do masks and hand washing, as much as you possibly can in the absence of a vaccine. Okay, so now if we have a vaccine, right, I'm going to look at an extreme case, which is when the vaccine is the only protection option. So we've abandoned all other protection options in favor of the vaccine. That's obviously not true because, of course, there are requirements to wear masks in some places, and there's, like calls for social distancing and so on. But I would say it does mimic a lot of the reality of what's going on. Right. So we looked at two options. One is when vaccination levels vary, and one is when everyone gets vaccinated. So obviously we're not going to see everyone vaccinated, but it's an extreme limiting case. So here's the case where vaccination is variable. And so the red is the risk before vaccination and the blue is the risk after vaccination. So obviously the risk goes down. That's not a surprise. But I think what's interesting is that there's a much greater spread here, right? And that's because the potential amount of people who are vaccinated can be quite large, actually, right? So it's between, we thought about 25% and 100% of people being vaccinated, right? And the parameter is varying simultaneously across this range. That's why you get quite a large spread. Okay, so if we look at different bands of vaccination, so what if it's very low, like 0 to 20%, or goes up, you maybe even have 80 to 100%, then you can see that the per day risk obviously gets lower the more people are vaccinated. Again, not a surprise. But what's interesting, I think, is kind of like the slope here, right? It's kind of skewed, and it's skewed. So that means if you picked a transmissibility level, right, you would run through a lot of different colors, whereas if you picked a daily risk, you would run through fewer colors, right? So what that means is the outcome is much more sensitive to the per day risk for a given transmissibility level than it is for varying transmissibility levels for a given risk. Right. Okay, so could vaccination result in a net increase in risk? Yes, it could if not enough people are vaccinated. So let's just look back at that for a second. All right, the previous one was about somewhere here. It actually would go up if you were in the purple zone. It would uniformly go up. And the reason is because you're abandoning things that are kind of working for something that nobody's taking. And then other people who are not taking the vaccine are also not doing anything. So obviously things are going to get worse. So yes, that's a clear yes in that case. But even some other cases, even if you have low vaccination, you can still make things worse if you abandon other protection options. Right. On the other hand, if you can vaccinate everyone, then you get a clear reduction, right? So that's lovely, but that's not going to happen in reality, right? So, well, we obviously need a large number of people to be vaccinated if you're going to abandon other protection options. So can we get a handle on how many and what exactly do we mean by that? Right. So here are the box plots of all the latent hypercube samples throughout. So this is sort of before the vaccine, as everything varies. This is when you have imperfect vaccination. And you can see that the average goes down for sure. The interquartile range also reduces, but it doesn't reduce that much, right? The range is still quite large. In fact, interquartile range is quite substantial here. Whereas if you can have perfect vaccination, well, the risk is quite low. The risk isn't zero because although you're vaccinating everybody, you don't have a perfect vaccine. So some people who are vaccinated might still have infection, but that's fairly low, right? The daily risk is now, less than 10% on average. All right. So if you don't have a vaccine, then the daily risk of infection is high. And imperfect vaccination only moderately improves this, but clearly vaccinating everybody, or in reality, a very high number of people, would reduce the risk to low levels. All right. So we next look at the sensitivity of the risk to the overall vaccine uptake, the vaccine efficacy, and you can see that the vaccine efficacy looks pretty uniform, right? So if the vaccine efficacy, is varying between 85% and 100%, the daily risk doesn't seem to change much. So as long as if your vaccine efficacy was lower than this, then it matters. But if the vaccine is reasonably high, which we now know the vaccines are, right, then it doesn't matter too much, with one exception, which I'll talk about in a moment, right? Whereas the vaccine uptake is a much more significant parameter. It's much more of a trend happening here. So the daily risk clearly goes down the more people who are being vaccinated. So that answers our second question, which is what's more important to maximize, vaccine efficacy or vaccine coverage? Well, we can use the vaccines we have because they're pretty good, right? And we're much better off getting those vaccines to more people than we are maybe improving the vaccine that we have. Right. And I said there was one exception. There is. I just want to mention what happens if you did vaccinate 100% of people, right? So in the ideal case of 100% uptake, right, then in that case, the vaccine efficacy has a very strong trend. All right, so this is where you vaccinated 100% of people. I'm sorry that the picture is overwritten, but the crucial information is down here. Right, now the vaccine efficacy matters a lot. Right, and you can see at the very corner, yes, if you had the case where 100% of people are vaccinated and with a perfect vaccine, yes, you would get the risk to zero. Right, so that's not too unexpected. All right, so the efficacy matters when coverage is high. Right, so we've kind of also answered our third crucial question, which is what fraction do you need to vaccinate in order to achieve greater than 50% reduction in transmission? So that was our previous curve. The 50% reduction line is this red one here. Well, if we kind of cut that off, you can see it's about 89% or so, right? That's not getting it to zero. That's getting it to half of what it was before the vaccine, right? And also, I just want to remind us, this is of all people, not just eligible adults. I think sometimes the stats are quoted to say, well, of eligible vaccinated people or something like that. But certainly in Canada, we see that a lot, but it's definitely this must be of all people, right? So that's a very, very big ask to get 89% of all people vaccinated, just to reduce the risk to half. That's a lot of people vaccinated for, well, it's a gain, it's a significant gain, but it's not everything. All right, so one thing we also want to look at was what I've called the bad parameter. This is the missing one, right? So because the protection protocol summed to 1, right, this wasn't an explicit parameter in our model, but it's in there. And so the bad parameter is basically 1 minus the sum of all the other protection protocols. Before vaccination, it's the PIs, there's 6 of them, and after vaccination, there's 13 QIs. And so if we look at the sensitivity to the bad parameter, then you can see it's sort of the opposite of the vaccine coverage. But what's interesting, I think, is it starts to hit 100%, right? The daily risk hits 100% if not enough people are using no protection, right? So they're not just not vaccinated, they're also not using masks or distancing. And after sort of 40% or so, you start hitting this ceiling, and that's really bad. Okay, so you get very bad outcomes because, of course, we're assuming a very extreme case here, which is that other protection options are being ditched when you've got a vaccine. All right, so if you had more than 50% of people not doing anything, then you're starting to talk about a really bad scenario. Okay, so the last thing I want to mention is the relative risk. And the relative risk is basically, can we say that the risk at least goes down because of the vaccine, right? So the outcome is beneficial if the risk before the vaccine is larger than the risk afterwards, which we would hope would be the case, but we've already seen it might not be if not enough people are vaccinated, right? And if we look at this threshold, or basically set them equal to each other, then we have this threshold here. A few things have canceled, and we can combine a few other things. And what we can see is, so here's the relative risk. We want to be above here. So the outcome is better if you're above. And certainly a lot of the simulations do give us outcomes above, but some of them give us outcomes below, right? So we can look at either moderate transmission or high transmission. And what you can see is that there's a worse outcome. It starts to get bad if there's not enough people being vaccinated. So less than about 40%, right? You start to see some significant numbers down here. Less than about 30%, it's almost 100% of the time you're going to get a worse outcome. Right. So I would say, if you can keep the vaccination rates above about 55%, at least you don't make things worse. And if you get vaccination below 30%, which is certainly happening in some countries in the developing world, you're basically almost guaranteed a bad outcome if you abandon other protection options. All right. So that's basically just summarizing here, right? And yes, I think we've done this. So yeah, I would say vaccines with less than 40% coverage are probably likely to increase the risk, right, in practice. All right, so let me just sum up. So we can definitely increase the risk if you don't vaccinate enough people, but you introduce the idea of the vaccine and then people change their behavior. And there's certainly something that happens, right? This is something that happens with STIs and so on. When they introduce vaccines and people think, great, I never use a condom again. And, the thing they're replacing is not always so great. Vaccine coverage can easily outweigh vaccine efficacy. There's a clear answer there. And if you want to achieve substantial daily reduction, by which I mean more than 50% reduction, you need to have more than 89% vaccination rates. Okay, so just a couple of limitations. We assumed uniform distributions because we didn't have a whole lot more information at the time. We consider masks as a single entity, but the effect varies significantly. That's actually true of other things as well, like distancing, but masks is a bit more obvious. So medical grade masks versus homemade masks are quite different in terms of their efficacy. And we just kind of considered them to kind of, be spread along the range. We might, for example, switch them into two different cases, like good masks and not so good masks. And of course, we were mostly dealing with an extreme case. Now, I didn't include slides where we looked at relaxing this extremity because we only had so much time. But in this particular case, you're looking at you abandon everything except the vaccine and hope everything pins on the vaccine. Okay, so vaccines do work, but you have to have enough people using them. Right. So the vaccine can theoretically control the epidemic, even when transmission is high, but you need sufficiently many people to be vaccinated, and you need to not abandon other protection options. And I think that's kind of the story to take away here, is because that's certainly something that has been happening. So I think the danger is that overconfidence in the vaccine with limited coverage could ultimately make the outbreak worse. So in conclusion, vaccines hold great promise of controlling the disease. We kind of adopted this first do no harm strategy, right? Saying let's at least not increase the risk because of the existence of a vaccine. And if we replace all existing protection methods with vaccination, that might not be the wisest choice, right? On the other hand, if we can vaccinate enough people, then we can reduce the risk, but we're probably going to need other protection options in the meantime in order to control the epidemic. Okay, so I'm just going to stop here, and I'm going to acknowledge my student, Stephanie Aber, who worked on this. And I would also just like to draw your attention to the date on this, because we published this in 2020, essentially before there even was a vaccine. That's the great power of mathematical modeling, I think, because we did most of this work before there was a vaccine, and then actually just as the article was being revised, the vaccines were announced. We didn't even have any data, but they were announced, so that we tweaked a few things as a result of the announcements. But a lot of this was stuff predicted, now, 2 years ago almost, well, maybe more like a year and a half ago, and much of it has come to pass in broad strokes. So if you're interested in more, you can read about this and other things on my website. That QR code will take you straight to my website as well. Okay, so I will stop the share, and thank you very much.
Lovely, thank you so much. That was a lot of material to cover in a short time. I appreciate it. We have time for a couple of questions. Not too much, but I'm sure there'll be some. So please go ahead.
I have a question.
Yes.
Oh, hang on. This has changed audio things here. Sorry. Question is accumulated immunity. So this seems to all work essentially in a entirely susceptible population. Do you have any intuition about what happens later on in an epidemic as we are now?
Yeah, that's a good point, actually, because you're right. As people both get the disease and then build some immunity, or get vaccinated and build some immunity that way. I think you're basically, what are you doing? You're taking out your susceptibles, right? So you have less susceptible people to then infect. So that would change the probability of infection each time. So I think you're right that, of course, if there is very good herd immunity, then you don't even need a vaccine. But of course, herd immunity requires immunity, and there's not great immunity from COVID. So I would say, that's actually probably another factor to put in, especially as we start looking at this disease as like a decade-long disease rather than a month-long disease, because I think we're living with this for a long time. And so, you're right. The longer we're in this, the more these long-term timescale issues come up. And I think that's an excellent one.
Thank you.
Other questions? You make a very good point, which is for a disease that has infectivity as high as the current strains of COVID, if you just do the classic herd immunity threshold, you get thresholds of 97, 98%.
Mm-hmm.
And so to get rid of it, you have to have extremely high, either everybody has to have had it or everybody has to be vaccinated. The vaccine has to be perfect. And so I guess one takeaway from this is that developing pediatric vaccines is probably pretty important.
Yes. Yes, exactly. That's right. If you have a major class of humans who can't be vaccinated, say under fives, then you're never going to get to these levels because, there's all the other adults who can't or won't be vaccinated as well. And you missed the herd immunity altogether. The other thing I wanted to note actually is when you say the classic herd immunity threshold, well, we actually did some other work on this, and that classic herd immunity threshold has never accounted for like multiple vaccines being developed at once, because we've never had that before. And you have multiple variants as well, which also changes it. So the classic threshold is a fairly limiting case. And so, once you start putting in other things, the herd immunity threshold also changes. So yeah, there's all kinds of quirks happening with COVID, partly because of the oddness of this disease, but also partly because of our reactions to it, which are in many ways unprecedented.
I guess, just thinking of classic SIR models, one case you're changing the beta, and the other case you're changing the fraction of susceptibles. Mm-hmm. And they, of course, they play into each other. And as you point out in your mathematical analysis, there's a lot of subtlety. But yes. but that herd immunity threshold estimate at least gives you a sanity check about what's reasonable. Mm-hmm. If the herd immunity threshold is 99% because your effective R naught is 10, then there's not a lot you could do.
That's right. Mm-hmm.
But I wonder what the lessons are. You focused on COVID, but this issue about I made a comment at one point unwisely on Facebook that one lesson from COVID is that we should all be wearing masks all the time, because we got rid of flu pretty effectively that way. Mm-hmm. I wonder if you could generalize a little bit to other serious infectious diseases and what potential generalizations there could be there.
Yes. So actually, we in fact did this work originally for Ebola, and we did it long before COVID was even a thing. And actually, what happened was we published it, and in the revisions, they said, "Why aren't you doing this for COVID?" And we thought, "Oh, because it didn't exist when we were doing it." So actually, we turned this around pretty quickly. And in fact, the editors went, "Yeah, you know what? You need to do this for COVID," and they were like, please hurry up because the vaccines are coming. And so, yeah, it's definitely something that applies more generally. Now, of course, Ebola, the vaccine isn't nearly as good. So you have further issues down the line. And we originally had done that thinking the vaccines would be potentially quite weak at first. And when they were announced, we kind of said, well, they're more up in the upper end. But if you have wider range of vaccination efficacy, then you have other issues coming on as well. And so, yeah, abandoning other protection options when you have a weak vaccine is even worse, of course. So I think that the more general takeaway is you need to think of these strategies as multi-pronged strategies. You can't just find this magic bullet. There isn't one, right? Yes, vaccines are great, right? But so are many other things. One of the funny things about COVID, I think, is I remember my colleague said, when it first came along, she said, should we get into this new disease? And I was kind of, I don't know, what do you think? And she said, well, it's kind of pretty boring, isn't it? And we're kind of like, yeah, it is pretty boring. It's a standard disease. Like, it's not quirky like HIV or whatever. Like, it's pretty ordinary, which means all the regular things work, like masks and distancing and quarantine, they all work. So great. That's cause for celebration in some ways. Now, of course, there are some quirks about COVID, but they're down the end of obscurity in lots of ways. Like I think there's this viral quirks, for instance, and so on. But in terms of its spread, like, we know what to do. We know how to deal with this. Whereas, previous things that turned up, we didn't always. So many diseases, of course, are going to have other quirks where they're not going to be quite as controllable. I think that the problem with this one is, like, because the herd immunity threshold is so high, it's high, but not like you say 99%. It's sort of 90% all, which seems like it's reachable, but in practice, it's a tough one to get to. And kind of trying to get everyone at once to do the right thing in the age of social media is probably a fool's errand.
But I think one other thing that you mentioned, I think the contrast with Ebola is interesting, and we use that as an example in when I teach, these kind of models in my fall class for undergrads. With Ebola, because the lethality is so high, people are pretty careful. And so Ebola epidemics tend to end, and there the vaccination can help prevent community spread, but people are going to be careful. Whereas, and if the lethality of COVID were a tenth what it is, people would let it rip and not worry about it. And it's just at this nasty balance. You talked about these sort of balance points where the lethality is high enough, it's a problem. but not high enough that people take it that seriously.
That's exactly right. Yes. Yeah, I was in Senegal when the Ebola outbreak hit, and it was fascinating. It was kind of like, baby's first pandemic, right? Watching everyone around me just immediately social distance. And it was like, oh, right, okay. And it was like, oh, yeah, we don't go into stores anymore. Like, we put our money outside, we walk away, they do things. And it was like, of course we do. And everyone is being very careful. And it was fascinating to me comparing that to March 2020 as COVID is taking off. And, in Canada, people going, well, what do? We don't know, and it was sort of like, you can tell who's been through a pandemic before and who hasn't, because, and when you've watched your relatives and neighbors drop dead, you take this thing seriously. But in COVID, at least for the first couple of years, say, we weren't really doing that. I remember, there's lots of those viral memes saying, do you actually know anyone who's had COVID? And people saying, oh, well, not directly, but, the numbers are there. But when it becomes, yes, grandma died of COVID and, so did my neighbor, then it's a different story.
Okay, other time for one last question for our speaker? No. Well, thank you so much. We really appreciate you coming and speaking to us. I hope you'll come again and give us another.
Yes, I'd love to. Thanks very much for having me.
That was very kind of you. So we appreciate that very much. And we will now move on to our second talk for today. We have Vivek Kapur from Penn State. And I don't have your title, so it'll be a pleasant surprise. So I'll turn it over to you.
Great, thanks much. Actually, I think there's another screen being shared, so I can't. Brilliant, thank you. So thanks much for inviting me to this. So first up, I'm not a modeler. I don't do any multi-scale modeling. I am a veterinarian by training, interested in molecular epidemiology, but have used quite a bit of multi-scale modeling in the areas that I work in, primarily in spillover and understanding spillover risk. And of my primary projects are in mycobacterial diseases, anthrax, brucella, other zoonoses, and like many got sucked into COVID. I'm going to share with you a story. It's a quirky one about Bambi and getting COVID. And it's something we stumbled into, weren't really looking very aggressively at, and it was sort of quite accidental, the whole thing. This is done with my colleague, Suresh Kuchipudi here at Penn State, who helps run the animal diagnostic lab, and we've got a fairly large many hosts of SARS-CoV-2 team. So let's speak with you about white-tailed deer. So Bambi allegedly is a white-tailed deer. There's some controversy about that, but Walt Disney really had anticipated and threw out the models of Bambi as a white-tailed deer. So white-tailed deer are the most abundant large mammal, depending on if you exclude humans, of course, in North America, around 30 million of them in the US. It's a fairly large economic impact, primarily from activities related to travel, to hunting, sightseeing, tourism, and there's a fairly decent sized cervid, farm cervid industry as well. This makes an interesting species because of their herd size and dynamics, their social structures, sort of seasonal influences in their range, their habits, their behaviors between breeding season, winter, depending on which part of the continent you're on. And all of these can impact pretty dramatically transmission dynamics. Fairly high density, this is sort of a density map of white-tailed deer and where all they spread. So primarily around the eastern and southeastern areas, but high concentrations around the Great Lakes area, now spreading over westward. This is actually a picture from my backyard. That's a really old peach tree, delicious old world peaches it gives. These deer just come hang out, grab the peaches. So they're a real pest, very, very annoying. I absolutely hate them. They eat the peaches, they eat the pears, and leave no roses for us at all. They reproduce pretty impressively. So those can drop twins, triplets sometimes, and you can have between 30 and 80% replacements in some herds, which are all very dependent on feed and nutrition, etc. Predators, at least in our area, we've got some wolves, etc., but are pretty far between, and it's human hunting that actually is the major form of population control in there. And I've seen deer drop in literally outside my garage and in the front yard. And being a veterinarian, I have to look after them, but I have to tell you, I know it's coming back eating our fruit. The interesting thing is after these deer eat this fruit, you can imagine it drops a whole bunch, and then the groundhogs come in and the squirrels come in, and then I'm chasing it around. And so the dogs and cats are all over the place. It's a pretty impressive environment with these deer in very domestic settings. I want to share with you a story about how we jumped into this. So during the fall and winter of 2020, we find that there was a silent outbreak of SARS-CoV-2 that was spreading in white-tailed deer. And this is a map of Iowa. You can see I've got sort of the parklands, green areas marked out here. But what I'm going to show you is things that are going to come up in as black and negative and red and positive of samples that were collected as part of a chronic wasting disease surveillance that's ongoing. So during the season, starting sometime in April, all the way through about March of the next year, which encompasses a big part of hunting season, and I'll share with you what happens there. Deer are sampled, their retropharyngeal lymph nodes are taken, and they're sampled for chronic wasting disease. And those are the samples that we started looking at. Samples collected between April and August are mostly sort of roadkill, very animals that are targeted because they're ill or behaving oddly, etc. So very few number of samples in this pilot set we looked at, and all of them were negative. And you can see it's a really, really small sample set. First positive in this set we got was actually in September, but subsequently we've seen samples as early as August, and it showed up in two parts. This is Sioux City, Iowa, an urban setting, and then right out here in the southeastern corner in an area called a preserve, which is a few hundred acres with a lot of deer, where people come in and shoot or visit those deer. So I'm just going to zoom into this area. You can sort of see amongst a whole lot of negative animals, we found this first positive one, and you can sort of see this is about a 10 kilometer sort of a square. If you zoom into this other hunting preserve, you see it's a relatively small area with the animals out. This is what happened in September. It's what happened in October. And that's what happened in November. And by the time December comes along, pretty much everything is looking positive that we were picking up. And then when you zoom out and you sort of see that there is about a third of all of the samples in this small set that we looked at were positive. And that to us was really stunning. It was truly gobsmacking because remember, we're looking for PCR of virus from lymph nodes in deer. So we have to have the right time. Just imagine if you're cross-sectionally serving a large fraction and you're getting 30% positivity rates. That's just something that was quite stunning to us. So this then would make white-tailed deer the first free-living species, free-living animal reservoir, non-human animal reservoir for SARS-CoV-2, which is a potentially big deal because, again, as I mentioned, deer is the largest large mammal in the United States. So this is what happens when there are none of those interventions that Stacey just mentioned. So no mask, no social distancing, definitely no hand washing, and there are no vaccines. And so this is sort of an idealized scenario for a virus, right? You're getting into a population. These deer are asymptomatic, so presumably you're not removing susceptibles through mortality. The trends are pretty impressive. I'm going to walk you through a couple of things out here. So first, in this light blue histogram out here is showing the number of weekly cases in humans in Iowa over that same time period, so starting April, ending March. And you can see there was a peak around November, mid-November, when there were about 1,000 cases per 100,000 population weekly, so about 1% or so. The first cases in deer start in about September. October, it's pretty low. Starts to rise in November, but in a short time, we're looking at around 70 to 80% of the deer were ending up positive in the samples we were taking. And out here, we're starting to take some fairly large numbers of samples. So that is quite impressive because we're talking about an order of magnitude of perhaps 2 orders of magnitude larger, greater than the case rates, if you sort of were looking at that in humans. So this was spreading pretty quickly. And again, when you have exponential growth, unfettered in a susceptible population, this is what one might expect, but still it was quite stunning to see it in real life, play out in real life. Just to say that the amount of virus that we were finding in these lymph nodes was not trivial. So this is not just sort of saying, oh, if you blink and if you squint, you're going to see a copy number. Some deer had up to 100 million copies per mil in lymph nodes. That is stunning. Again, quite shocking because these deer are asymptomatic. So they can apparently harbor a lot of virus, presumably spread a lot of virus, and yet there's none of the cytokine storm that or other signs and symptoms that humans get. We of course, sequenced, did whole genome sequencing for each one of those isolates that we picked out. And what we see is very clear evidence of multiple spillovers. So what you're seeing in this salmon color versus in gray is the background of what the cases were present in Iowa. So this is looking for whole genome sequences deposited around the same time period from humans. And then you can see these sort of spillovers occur in deer. So you have this human case, a spillover into deer, and then replication within the deer, spread within the deer. And no matter where you look at it, you're seeing the same. And so depending on which lineage there was circulating locally is pretty much the lineage that's showing up in deer, again, suggesting that this is a spillover from humans to deer. That's followed by then within deer herd transmission. So how might this happen? There are a variety of ways. Contaminated food sources. So if I were eating an apple and threw it in my yard, or a kid throws it out of the window, a deer picks it up. That's an easy thing. And then given that deer are so susceptible, it's gonna whip right through. Contaminated environments, somebody taking a walk in the woods, hiking, peeing, pooping, are the sorts of things that would also allow contact between infectious material and deer, and that would do it. Or we can't even rule out an intermediary host. Is there a feral cat or a deer mouse, perhaps, that harbored it and and made contact with the deer. So we've gone back in and asked questions. I mean, this is not all that many samples, and we've got a lot more that we've got now. But when you look at the samples from deer in this area, there's of course a very strong temporal clustering. In blue out here are samples from human. In this orangish color, salmon color, are the samples from deer. You can see pretty strong temporal trends. And then if you sort of do straightforward analyses looking for the time for the last most recent common ancestor, we get right around the beginning of the pandemic. So the first case in Iowa was March 8th or 10th or something like that, and we see the origin of those sequences in 2019, early Jan, and then around 40 days later, you can sort of see the deer sample origin. So if there was anything, it was a pretty quick spillover. When you run transmission network, sort of transphilo like analyses to look to try and look for transmission networks, who gave what to whom. Clearly, there are a lot of missing cases. Deer appear much better sampled than humans. And again, very clear evidence of multiple spillovers from an effectively human route into deer, again in the salmon color, as opposed to the blue that you see in humans. We do not see any evidence yet of deer to human spillback. We see no reason why this might not occur. But indeed, there is no current evidence of that. Regardless, the data are pretty suggestive of deer being a new reservoir host. And of course, that's important for a variety of reasons, particularly under control of the pandemic. I just want to this is data after we published our paper, just came out in PNAS. From that initial survey, I mentioned that there were about 5,000 samples. This is about, we just jumped through around 3,000. And this is what the picture actually looks like. It's really, really stunning. And because we have sequences from all of these, we've got now at least an order of magnitude more sequences from deer than we have human. And given the fact that there's none of the behavioral factors that would influence transmission, we have a really powerful means to go in and investigate what exactly happens when this particular virus spreads into a susceptible new host and rips through it. So it's actually quite fun. It's working out to be quite a fun side project for us. I just want to end with a couple of general thoughts. So the reason why we looked at deer was because there was good evidence from either experimental or computational predictions that deer, experimental studies, so actually challenging deer, that deer were indeed going to be susceptible and they were going to be able to transmit this. There were trials not only with the experimental infections, but also serological surveys that suggested that deer were converting. So about 40% of the deer in some of the early surveys had evidence of antibodies. Most of us said like, I wonder where that's coming from, and forgot about it. And we didn't really look very seriously for the virus because we had sort of a very human-centric mindset, thinking that, if you go do a nasal swab or you look for the virus in a human, you've got a short window. What's the probability of finding a deer and they're asymptomatic? What's the probability of finding a deer that has a virus in it. So it's the serological screens that are going to give us a good look to see how this infection is spreading. So we were sort of shying away from looking for the virus actually in these because we were very busy with doing the human SARS-CoV-2 screening, and so just as we got a gap is when we got into this. But when you actually put down all of the animals that have been either predicted, shown to have either natural infection or experimental infection, or are predicted based on just computational modeling of ACE2 and spike binding, whether they have clinical signs of virus shedding or transmission, actually see a very large fraction of animals have something or the other reported. And many of these also have potential for spillback risk. And that should be of no surprise to us, because if this thing came from a bat, as we suspect, with or without intermediate hosts, humans managed to do a fantastically good job spreading this virus around, including spilling over into mink and then spilling back from mink. We and others have shown this spill over into deer. And there's no reason why this is not spilling back into multiple other species. Livestock, we haven't really found evidence, others haven't, though we can't rule it out with what might be going on with new variants. And so that's something that we've got a keen lookout for. And then there's lots of reports about companion animals as zoo animals in captive settings that get presumably pretty high infectious doses or frequent exposures, pick it up and then transmit amongst themselves. And no evidence yet, including from Hong Kong. We've been looking at this hamster story. It's just unclear what the evidence is of spillback from those settings into humans. Humans are the biggest risk to humans at the moment, but they're also a very big risk to animals. So I think one can create these plausible transmission networks, which are really multi-host systems where humans give it to humans, put it into other animals, which can then start their own transmission networks, and then go into bats and come back in different shapes and forms. And this, of course, was the concern when we started first looking out, asking the question, are there many reservoirs? So many of us have sort of resigned to SARS-CoV-2, ending up somewhat like this other familiar pathogen flu, of course, with different hosts and different dynamics. But of course, we understand there's a lot of recombination that occurs in coronaviruses, including the SARS lineages, and there are quite a few endogenous endemic coronaviruses in things like deer and other free-living species. So there are many open questions. We're sort of asking them now, and a lot of our colleagues are chasing this down as well. So will it burn out? If you get such rapid spread, do you just get fade out, or does it become endemic? And so we're keen on figuring out what really R0 might look like, and what happens with repeated cycles of new recruitment of susceptibles, with births and elimination. Does this adapt to the host, and then does it make it nastier for other species potentially, or milder, better? What's going on there? And so we're, of course, expanding surveys, doing more sequencing, etc. We're very keen on looking at spillover and spillback risks. We're keen on trying to better understand the ecological scale of transmission. So what happens? And for that, we're initiating longitudinal multi-host surveys. Really curious as to why deer with such high apparent viral loads stay symptomatic. And so there's certain mechanistic studies out there. There's a lot of captive surveyed. So the question is does vaccination have a role? Or is it just a tool we can use to understand what vaccines do in the absence of the hand washing, masking like scenarios? And we're very keen on understanding what this might mean for the end game for the current pandemic. Again, I'll just sort of do a plug for One Health. In fact, this clearly shows how that interconnection exists, and a need for better coordination and integration of approaches for disease surveillance in general. And I'll close with that. That's a paper that just came out in PNAS. I'm happy to share that with you. And the work was done with lots of colleagues all over the country, and in the UK as well. And we've been really a really good team that stood up in a matter of weeks. So September 30th was the first time we got a positive in a deer sample. And by December, we had a paper accepted, but pretty wide analyses done globally. So I'm going to stop there and happy to answer any questions.
Okay, thank you very much. That was extremely stimulating. Questions for our speaker? Well, maybe I'll get started then. You mentioned that the deer are apparently asymptomatic. And we had a very intriguing talk from Claire Bryant a few weeks ago from Cambridge, talking about the differences in the nature of immune response in different species. And one of the things that she brought up was the fact that there are certain cell death mechanisms that are missing in certain species, which can lead to situations in which a particular virus or bacterial pathogen will not cause significant illness, but won't be cleared. Now, if I understood you right, all of your sampling is ex vivo sampling. Is there any way to infer, are the animals staying infected? And if so, for how long? How long does the viral load stay high? Are they clearing the virus? Do they have any immunity? Is there any way of even getting rough estimates, maybe from your modeling of the transmission based on the data sets you're collecting?
Yeah, thanks for that. So the samples we're looking at are retropharyngeal lymph nodes. So these are the lymph nodes that drain pretty much everything in the upper respiratory tract. It's the same thing we use for TB or for other things, right? The experimental data, when you actually experimentally infect a deer with SARS-CoV-2, suggests that there are two areas in a deer where the virus tends to persist a little bit longer. And that's the palatine tonsil, and the other is the retropharyngeal lymph node. So for around 20 odd days, 28 days, depending on one study, 21 and another, you could actually get PCR positive virus out of these. And then if you put sentinels alongside those experimentally challenged animals, the period of infectiousness, so where onward transmission might occur, is a period between 3 and 6 days. So it's a pretty narrow window, but the virus hangs around, but is presumably not infectious. I think we might want to uncouple what we're seeing in those experimental settings with what might be happening out in nature, primarily because we have no idea when deer are stressed, what impact they might have. And arguably most humans are relatively asymptomatic as well. Deer, when under nutrition or other stressors, might actually behave pretty differently. So we just don't know that yet very well. I do think, though, that point about the sampling is really important, because if you take a nasal swab, the likelihood of that getting positive is pretty, is much lower than if you were to take this retropharyngeal lymph node sample. But then in a way, what the retropharyngeal lymph node acts as is a fraction of those would be infectious, and a fraction of those would be recovered, a fraction of those would be just newly infected, right? So we have to then, if you are putting it into a model, you can't, an SIR model would way overestimate what R naught would be. You have to account for that, which is really important to do. And so that's sort of the way doing this. Having the genome sequences has been hugely important because their family structure and their herd structures are, 15 to 30 individuals or so. And then you have males that visit during rut season. So we know that sort of mixing. We know that their home ranges are not a lot. And because we've got GPS coordinates for where the samples were taken, you can actually see the patterns. So much better than anyone could ever do in terms of where they might have got infected, how the infection spread, how many times the spillover occurred. So I think we've got pretty good handle on that part.
So I guess I didn't realize that so these experiments are being done in captive populations then.
They have been done as well in captive populations. So our studies, of course, were entirely mostly free living deer, but some deer were in these preserves, which are in managed settings, where there is some more interaction because they're nutritionally supplemented.
But I guess and I want to let Ruchira ask her question, but just to finish this. So, but the evidence is in these captive populations that the deer do clear the virus.
Oh yeah.
And is there any evidence that there's any immunity after that first clearance? Or their duration in their interval to reinfection?
Yeah, so those studies were not done, but you get very similar sort of trajectories for antibodies, CMI responses in India as you do in humans or other species. We see very clear evidence of rapid generation of IgG and M almost simultaneously, like you see with any rapidly replicating virus like this. And then we see the memory cells show up relatively quickly as well, which is quite impressive to see. You can see pretty good neutralization with antibodies. You can run some VNs, and it's pretty clear what we're getting. So antibodies neutralize. We've not done any CMI sort of detailed work there, but one would expect that you get good responses. Yeah. And you also get vertical transmission. So we've seen it go from a doe to her unborn fawn, so a fetus as well. So there are other transmission mechanisms that could be there. Yeah.
Ruchira, thank you for being patient.
Sure, thank you for organizing. So thank you for that very interesting and stimulating talk. So I have two questions. One is, to what extent is it known, especially maybe it's known from the captive studies, whether the SARS-CoV-2 stays in other organs in deer, like in the gastrointestinal tract? So that was one question. And the other was, so given that it seems to be circulating and present at a high rate, have you thought of or tried tagging deer so that you could catch and release and follow them longitudinally, the free living deer?
Yeah, thanks.
So you would get the same samples from the same deer multiple times.
Yeah, no, thanks for that, Ruchira. Great question. So I think let me take your second question first. So from a big picture perspective, those longitudinal surveys and age-structured surveys are really critical, and that's what we're starting to do right now. There are some places where animals are tagged and are resampled, and we're going back in to do that very thing that you asked. And I think that's absolutely essential if we need to understand what the long-term trajectories and whether this becomes endemic or persists in this population, or otherwise you just need an exogenous source of spillovers all the time for it to persist. In terms of other tissues, people really, so pretty much all tissues were looked at. It tends to not get into the lungs really well, stay pretty much in the upper respiratory tract a lot, and lymph nodes, so tonsils and lymph nodes. It has PCR positive in poop, but no virus recovered from poop yet. It was something that we were very interested in because, that gives another transmission mechanism and persistence mechanism to other species and sort of potentially back to humans. But we've not seen evidence of at least the strains that were early on looked at in those experimental systems, right? Because these are also extraordinarily variant dependent. So the only strains that have been put in thus far have been the early Washington-like, so sort of the old Wuhan type, and then Alpha. Delta has been recovered, Omicron's been recovered, but it's not been put back into deer yet. I would predict, if I were betting, but I've been so darn wrong with pretty much all my predictions at this thing. If I were predicting quite foolishly, I would say that different variants would behave quite differently in terms of organ tropism and likely persistence in deer. Yeah, I should stop predicting. Yeah, I'm just so bad at it.
That's okay. We won't hold you to anything. Thank you. Did you have another question, Ruchira? Or?
No. Thank you for going out on a limb.
Jim, did you have a question?
Well, he partially just answered it. So you don't see virus in scat, but you do see a PCR signal?
Very early on, and we've not recovered any virus. But again, remember, these are well-nourished deer in experimental setting with one dose going in intratracheally, right? Or intranasally, or depending on who did the experiment. In natural settings, if you're out in my yard now in winter, you can see these plumes come out of deer. Mm-hmm. They're breathing hard, and you can see exactly why they're spreading this thing. But if I'm suspecting, if you've got deer interacting a lot with repeated exposures, there's very different dynamics going on, and that's not been yet examined. We have deer pens here at Penn State, and part of our work is to go back in and look to see when spillover might occur, or if it occurs, and then how we might be able to track it. Yeah.
Okay, thank you.
Other questions? Maybe following up on that, do you see any evidence of superinfection or co-infection when you do your genomics?
Yes, so great question. Our colleagues at Kansas State actually did that experiment. So they infected with Washington, so sort of the root lineages, and then chased with alpha, and see that alpha actually outcompetes in vivo. So you put in a bit of both, and you get out mostly alpha out. So there's no reason to suspect that that's not happening in nature with multiple. In our studies, when we're looking at these sequences from tissue, we do see minor allelic variants. So a lot of minor variants show up, but there is just whopping amounts of a dominant strain in there. So we've not seen it yet, but clearly experimentally, you can reinfect. You can super co-infect these things. Yeah.
So maybe for those of us who aren't specialists in zoonotics, are there other human diseases that white-tailed deer are a reservoir for that are well known? And what is the spillover pattern of those, if known, compared to that for COVID?
So my real work is in tuberculosis, and so deer are indeed a reservoir of TB, and there's spillover to livestock species, and as well as reported into humans, and humans giving it to deer as well. So there's just a lot of spillover, spillback with with TB complex organisms. We also know about chronic wasting disease. Again, prion base, so that's TB is a bacterium, chronic wasting disease. And then there are other viruses that are of more relevance to livestock populations, like EHTV. So just a whole bunch of other things that go in and out. Other coronaviruses you can recover quite easily from deer. Those are mostly the ones that are the bovine type. So between livestock and deer populations, you can get quite a bit of multi-host transfers. Yeah.
Okay. Reed, please go ahead.
Hi, yes. Yeah, very nice talk. Sorry to join a couple minutes late, so apologies if this question's been covered. I was looking at your phylogenetic tree at the end. I was curious if anybody has done like an overlay of ACE2 sequences from those animal hosts to see if there's any, how well that matches your observations here in the literature and otherwise. And I guess comments on, could this be ACE2 independent as you get outside of primates? Thanks.
Yeah, thanks. Great question. So yeah, so ACE2 predictions were the first ones that suggested that white-tailed deer would be highly susceptible, and that's why people did those experimental infections, because you sort of looked at it and it was amongst that high, high predicted susceptibility. But then mink were the low ones. They were thought to be less, sort of the least susceptible based on the computational predictions. But yet mink turn out to be, you just need one little switch and the virus happily then works out in and spreads in mink. So when you're doing just, there's about 400 or so ACE2 sequences in the databases from different species. And when you do those sort of computational predictions, ad hoc, I mean, just pretty much anything you throw at it, it's reasonable, but it's you get these exceptions like mink that really throw you off, because there are two species, deer and mink, that have been shown to have very, both very dramatic levels of infection in them. It gets into the mink population, it spreads like crazy, and all mustelids, in fact. But yet the predictions were that it would be very poor. And then you've got deer, which high predictions, and it does a really good job. And then if you look at just the primate component in this tree out here, it's pretty good out there. But then you've got these oddball things where you're sort of looking at and you're seeing good predictions computationally, but then other animals close by, wood rats, others, might actually have good experimental infections in them as well. So just depending, if you want to look at that lens, what level do you want to look at out here? So the Egyptian fruit bat or the big brown bat, very different in their susceptibilities. But based on ACE2, you wouldn't know that difference.
But
Great, thanks.
okay, other questions? Maybe following that up a little bit more, you mentioned that in deer, the infection seems to stay upper respiratory and lymph node, and not so much in the lungs, which was, of course, and I guess you don't have those kind of data yet for Delta and Omicron, or is this, so this is still mainly for the earlier strains. But I was going to ask a question. You talked about tuberculosis as a reservoir in deer, and certainly when I was thinking of co-infection, and I was thinking primarily viral co-infection, but certainly viral bacterial co-infection is important, and tuberculosis could certainly have all sorts of complicated effects on immune response and susceptibility. do you have an estimate of what fraction of the deer have are TB positive? And if so, do you think that the tuberculosis in the I assume in the captive strains, it probably is, I don't know, is it lower rate of infection? And if so, do you think that the presence of tuberculosis in the population could be affecting the spread of the virus?
Yeah, no, great question. So I think we're very lucky that TB is not very widespread in deer, and there are only certain parts of the country in which you can see that, and it's reported with frequency. So Michigan, Wisconsin, Minnesota, and then some other areas. I would actually worry about other bovine respiratory viruses. There's just a whole bunch of them. There are other bacterial pathogens that are respiratory pathogens in many ruminant species, sort of broad. The Pasteurella like organisms can cause havoc in deer, and in fact, co-infections are extremely common. Much like in humans, a primary respiratory virus leading to a secondary bacterial infection are pretty common in deer as well.
Thank you very much. Any final questions for our speaker?
So I just close by saying this thing is full of surprises. And the one thing message for us was, if we hadn't been looking, we actually would not have found this. And it was completely opportunistic for us to look at these samples. And I'm just wondering, and Ruchira has a few comments around Lyme disease, which of course another reason why I don't like deer, because that's exactly what those guys gave to me. There's lots of potential networks that can be and should be looked at. And my greater worry is that this is endemic India simply because of us ignoring to look at those networks. I think with an R naught or whatever we get on this one, it's still gone. It's nothing we could do about it. There's no intervention. You couldn't remove enough of the deer to do anything about it. There's no strategy to immunize them. So, when I look at this, what I really, really wonder is that how might this persist? So what's next year going to look like? And already I can tell you this year, we're finding a fairly decent chunk of the population you can recover virus from. And indeed, including in animals that might have antibodies. So we just think, we're going to have to live with this one.
So you are seeing evidence of reinfection then?
We suspect, yeah.
And why is vaccination out of the? Is it a cost issue, an availability issue? Why would, or what we heard about in the first talk, which is that the level of vaccination you need to make a difference is so high that it's impractical.
I think in captive cervids you could vaccinate everything, and but again, as we're aware, you want to ask, what is it that you're trying to stop? If you're trying to stop transmission, then we know that the current vaccines are not very good at that. And it doesn't matter what 1 - 1 over R naught into efficacy formula you use, it ain't working. At 100% coverage, you're not blocking transmission. Disease is not a big issue because there are no signs. So what is it we're trying to actually accomplish here? And in free living animals, it's just not possible. Yeah.
Fascinating. Well, thank you so much. We really appreciate your coming to speak to us. And I think that it'll give people a lot to think about in terms of how to apply these ideas to modeling. We've had a number of talks on zoonotic relationships, and they're really important, and we don't hear enough. So thank you again for coming.
Thanks for the invitation. Lovely being here.
Okay, with that,
Bye for now.
good night. Call the meeting to a close then. Thank you, everyone.
Thanks.